Okay, hi everybody who's joined up until now. We'll give people another minute or two to file in and then we'll get started. Okay, hi everybody and good afternoon. Welcome to the first Bay Vision virtual meetup, AI medical imaging. Hope everybody that is doing well during these challenging and crazy times. We really appreciate the amazing response we've had to this webinar. We've had well over 300 people registered, not only our usual community from the Bay Area, but really from all over the US and all over the world. And really heartwarming and great to see the names of some people we already know, who we've met before, and really appreciate everybody's participation. So this is a Bay Vision virtual meetup, AI in medical imaging. My name is Moshe Safran. Today I'll be hosting Jagadish Venkataraman from Calico Life Sciences, who will talk about from endoscopy to microscopy, the role of machine vision. A few words before we get started about who we are and about our meetup. So we're RSIP Vision. As many of you know, we create and provide computer vision solutions for medical applications. And we started this meetup a couple of years ago in order to share our knowledge and enhance our connection to the local AI ecosystem here in the Bay Area and beyond. We hold these meetups usually once every month or two in Cupertino. Usually actually during the evening, people come in and it's a networking event. Traditionally, some of the time we speak and share our experiences. Other times we have guest speakers from industry and from academia. And it's a really great event. We believe as a company, both internally and our relations with the outside world, really people are the key. This is how we motivate ourselves as a company, and we really are happy to share this passion, both for the technology and the mission to use it for sort of a wider good. Really happy to see that this new online platform for continuing seems to be taking off quite nicely, and we'll be happy to do more of these down the road. As an AI-focused company, computer vision-focused company, we work really across verticals and across modalities in the industry. And of course, as we all know, we're seeing a very wide-ranging and increasing role for computer vision and AI across the medical applications and verticals. Obviously, at this time, there's a lot of flux in our space and everywhere else. So ranging from people who are saying, yeah, our company is now massively diverted to COVID, to software houses like ourselves who are currently mostly unaffected. We're working remotely, transitioning, etc. Others are using some time or some resources that have freed up to plan their roadmaps or to increase their ground truth data sets. So there have been various responses, and we're going to see in the coming months, obviously, how our industry is affected. We're obviously going to be touched by this current situation very significantly. Nevertheless, we all know that the need for continued automation in this space is as pressing as ever. So workload of radiologists and physicians is not going to go away. The need to make planning times for procedures quicker and more efficient, precision, safety, all of these needs are still there, and we are all happy to be a part of the community and the ecosystem that's contributing. Just to share a few recent examples before we go to the lecture of our work. Again, we're really working in many different fields. On top, this is an example from endoscopic video, doing action recognition, image segmentation, and motion estimation, in this case for a GI application. I think Jagadish will talk a lot about surgical video, which has many use cases for computer vision at many stages of the procedure. Ultrasound analysis, we're seeing also strong interest, classification tasks, segmentation tasks. This particular example is estimation of fractional area change, but there's a ton of room in this modality for AI and for automation. The bottom middle row, you can see a new 2D to 3D reconstruction and image segmentation end-to-end network that we've put out, and are actually we have a paper currently under peer review. We also work in microscopy, mainly on nuclei and tumor region segmentation and cell classification, and the list goes on and on. Really, the uses for computer vision in medical are really endless. We feel quite fortunate to be part of this community. Now, our guest speaker today, Jagadish, I met him at the AI Accelerator Summit back in February down in San Jose. We were both speakers there. It was turned out to be one of the last opportunities to meet others from the community at this type of event. So Jagadish really is a consummate professional, and really immediately I felt that we have this shared passion to use this technology and to use these capabilities for a greater good. Jagadish holds a PhD in electrical engineering, started his career in semiconductor and navigation applications, and then decided to transition to healthcare and life sciences. The start of his career was at Verb Surgical, where he laid the foundations for perception targets for their robotic surgery platform. And he is currently at Calico Life Science, devoting his time to automating and scaling up analysis of high-resolution microscopy images with the goal of further understanding and solving of aging-related disease. So I'm very happy, Jagadish, to be hosting you today. And without further ado, I will transfer the presentation to you, and we're all looking forward to hearing your talk. One last comment, towards the end, we'll open it up for questions, so you can type them in, and we will try to find time to answer as many of them as possible towards the end. Okay, so Jagadish, the floor is yours. All right, you got it.
All right. Do you see my screen?
Yes.
Okay, cool. Hello, everyone. Thanks for coming. I really wish we were meeting under different circumstances. Yeah, but these are crazy times. And thank you, Moshe, for organizing this. So, yeah, I just wanted to sort of have this as a sort of distraction from everything else that is going on, so that we can focus on something that we are more passionate about. So the title of my talk, endoscopy to microscopy, role of machine vision, really sort of summarizes things that I have been looking at for the past 3.5 years or so. Sort of one of the motivations of my talk itself was something I heard at a MICAI conference a little over a couple of years ago, where the chair of the conference essentially was saying that over the past 25 years, we have had over 2,000 papers published, sort of promising over the promises of AI, and it's coming, and OR is changing, and diagnosis is changing. But he could literally count on his fingers the number of those papers that actually got translated into deployed solutions, both in the OR and in the clinician's office. And really, so the way I'm sort of structuring my talk is to sort of approach it more from a data and modeling perspective, sort of motivate the use of vision, computer vision, and image data in an end-to-end analysis, and also try to sort of see what is lacking and what we can do as a community to sort of step up and make things happen. So Calico is a part of the Alphabet family, and so the work, the material I'm showing here sort of is a union of work that I did at Verb, Alphabet, and Calico, but nothing specific about the projects ongoing at either of those places. A very generic talk. All of the material here is from publicly available papers. The links are there in the slides for you guys to go through and sort of go through them. The only thing that I will say related to Calico is on the last slide, where I have a little plug for one of the computer vision applications at Calico. All right, so with that caveat, so I'm going to sort of talk about vision in the medical space first, and then transition into biotech. So I want to sort of start with how vision has come to play a key role in surgery, and then what are the different kinds of applications people are looking at today, and then we'll go from there. So the first slide I have is, let's see. Okay, so this is how many of you might have already seen this. This is how surgery used to be performed in the early late 19th century, or early 20th century, and so it really was a theater, and you had people literally crowding in and seeing it, and most of it was open surgery, as you know. And then with the advent of laparoscopic surgery, with improvements in video quality and like cameras and the miniaturization of cameras, really the surgery transitioned to instead of cutting open the patient, you had the ability to now just have 3 or 4 incisions in the patient. One of them was reserved for a camera with a laser light source, which would essentially sort of transmit to the surgical team a feed from inside the patient. And then the other ports were essentially used for the tools that the surgeons and their assistants would use to sort of navigate through the anatomy and get the procedure done. So it was a clear win in going from sort of open surgery to laparoscopic procedures. It was surgeries were much faster, much lesser bleeding, much faster recovery times, costs were lower. So overall, it was like a win-win scenario when transitioning from open surgery to laparoscopic. And it really took off in the procedures where it was possible. And then came robotic surgery again at around 1999, about 21 years ago, where they introduced this robot, which is essentially positioned over the patient that you have here. And the surgeon, instead of being in the sterile field scrubbed in with the rest of the staff, he or she is actually at a console where they are essentially watching the video feed. So one of the arms of the robot is carrying the laser light source and the camera. And it's so transmitting the feed real time with very minimal latency, and there's heavy constraints on the latency, so that it's really real time for the surgeon. So the surgeon is essentially watching that video feed here. And they are typically wearing sensors on their fingers and making movements in the air, which are then mimicked inside the patient to essentially go and perform the relevant motion. So the only feedback they have in this kind of setup is the visual feedback that they're getting from the camera. So there is no notion of a haptic feedback. They cannot really feel the anatomy. They cannot feel how hard they are pulling and things like that. So essentially, it's reduced to what is called visual haptics. Essentially, they are just able to see what is happening, and then if they're tying a knot, just visually verify how tight the knot is, and so on. So, like I said earlier, transitioning from open to laparoscopic was a very clear win, but that really was not the case for, or rather, it's not as clear a win for robotic over laparoscopic, essentially because it's not like the times, the surgical times have gone down. In fact, if anything, the robotic surgeries may take a little longer with the overhead of having to set up and dismantle the robot and so on. And also, there was not like a big difference in, at least in some of the procedures, in terms of outcomes. So it wasn't it was incremental compared to what happened from open to laparoscopic. So having said that, it does introduce a lot of other dimensions of freedom for the surgeon in the way they can manipulate the anatomy and articulate some of their gestures. So it definitely sort of paves the way for democratizing surgery, in the sense that now you have a robot that is involved, which has so far been dumb, but can very easily now be sort of incorporated with perception and other mechanics to help the surgeon during the procedure. Okay, so that is sort of what we are going to be focused on. And then so this is sort of what people think of when they talk about the future. Essentially, they have like AR, VR, and what have you sort of integrated into the surgical robot. And there's also a lot of media hype about what is possible, and so that sort of creates a little bit of disillusionment, if you will, because the progress really is not happening at the pace we think it should be happening at, because the rest of the computer vision community is sort of exploding. If you look at all the apps that are coming out, and the advances made in the self-driving space and so on, but it's kind of been a little hard for the medical and to some extent the biotech space to keep up with. And so I'll sort of try and go into why some of that is there. So really, if you think about the entire patient experience from the moment they walk into a doctor's clinic to the point when they are diagnosed with a particular disease, and to the point where they have a certain procedure done based on what came out of the diagnosis, and to the point where they are discharged from the hospital and they have a bunch of aftercare, post-op visits and aftercare analytics that happen to them. So essentially, what is referred to as collective surgical consciousness. This is from an artificial intelligence and surgery paper that came out a couple of years ago. And as you can see here, in every box here, whether it's the diagnostic phase, the intra-op phase, or the post-op phase, you have some vision component that can play a role. So even though you don't explicitly see it here, so I'll come to that. So so what we will do is we will take up one example of a particular disease, and we will go through the diagnostic phase for that. We will look at what is the surgical intervention and potentially what can be done post-op with some of the images and videos that were sort of collected during the procedure. But the goal of this slide is to essentially say that collective surgical consciousness is the entire patient experience that can be sort of improved with the integration of imaging data, which today is under-integrated and underutilized in this entire chain. So the disease we will talk about is gallstones. So for many of you may already know, for those who don't, gallstones can range in size from, like little grains of sand all the way to like little golf balls. Almost, they can be cholesterol stones or, like pigment stones. And the first mode of sort of deducing them is through an ultrasound. Okay, so and if the stones are usually big enough, the ultrasound is pretty conclusive, and the next logical step is to essentially remove the gallbladder, infected gallbladder out, and the patient undergoes the procedure, and then they walk away home the same day. So that is typically the chain that happens. But there are often cases where, depending on the size of the patient, the size of the stones themselves, and so it's not very clear, and the stones may be like they may be causing infection, they may be causing pain, but looking at an ultrasound is not conclusive, in which case the next line of attack is a CT. A CT is it sort of gives you a little bit more, context in terms of the soft tissue and the blood vessels and things that are surrounding it. And then it may, and you're trying to sort of identify where are the stones. And as you can see here, highlighted in these little yellow blobs are the gallstones, which the radiologists typically have to stare at and essentially come up with a conclusion as to whether they're really stones and what is the next step, right? So the one of the papers that was looking at these CT images was to used a sort of modified version of a fully convolutional network. So you all may be familiar with the UNet, which is extremely popular in the medical community. So it's essentially like a fully convolutional network, sort of like an autoencoder model, where you can go from an image to a segmentation map that you wish to come up with, or it could be used for other purposes as well, as I will talk through later. But the goal here is to essentially go from here, say that, okay, let's say a bunch of radiologists have looked at these images and they have actually gone and colored all of these pixels and said that, okay, these are the stones. And let's say they have done these on sufficient number of images to train your model. The input is essentially the image stack. So CT can be actually three-dimensional as opposed to X-rays. So you can have a 3D image going in to your model. And the output essentially can be the segmentation map across the different Z slices that the radiologists have annotated, essentially. And instead of the traditional unit, the authors of this paper came up with what they call the UNext. Almost like a unit on steroids. They borrowed a lot of very generously from some of the DenseNet features, where they have like skip connections, not just to the corresponding layer, but they also have like skip connections similar to a DenseNet to every subsequent layer as well. And they also have little attention blocks. So what these attention blocks do is so in the unit, as the image sort of gets downsampled, so the image keeps reducing in size until it reaches like the bottleneck layer, and then it sort of keeps getting upsampled back to the original size of the image. These attention blocks, as you go down, try to focus more and more on the regions where there is signal, and the background is usually thrown away when you do the max pull operation. So by feeding some of these attention block information to the layer above, they have created this attention model where essentially they are able to tell the model where to look, so that you can sort of featureize it better compared to a traditional unit. So that is kind of the innovation they have in this paper. And then so this is an image, and you can see like a little white blob here, which is the gallstone. This is a segmentation map of that gallstone. So this is how a regular or traditional unit does, and you can see some false positives here, and you can see that even the gallstone that was annotated is broken up. So they also have a ResNet backbone fully convolutional network that they tried, and I believe that one that did slightly better than the unit, but not as well as their UNext model, which seem to do the best, at least in this particular data set. But having said this, is this model ready for use in a radiologist's office? I'm not quite sure, basically because the entire data set presented in this paper is about 5,000 images, annotated by only 4 radiologists total. And there is no mention of what is the inter-radiologist disagreement. So it's not clear if they multiple people annotated the same image, if so, to what degree their annotations overlapped, and so on, right? So this is a very tiny data set, and it's a good paper, but essentially to be able to take this and scale it up to like a production level model is a whole different ballgame. And that is really the bottleneck that sort of is hindering the community at this point. And then, okay, let's say that the CT did come back saying that, okay, here is a gallstone. Then the next logical step is to have the gallbladder removed, which in this case is a procedure called cholecystectomy. Again, a lot of papers published on this, a lot of data sets out there, not a lot of data sets, a couple of data sets at least out there. And there was even a talk about this at this AI summit that Moshe mentioned from Intuitive folks. So Intuitive has actually sponsored challenges with data sets. There is a data set called Collect 80, which contains about 80 surgical videos. So the problem that they're trying to solve there essentially is that the gallbladder removal procedure starts with the preparation of the patient all the way to the gallbladder resection. It's a very standardized procedure, which has about 7 steps end to end from the time the patient is open to the point they are closed, right? And so the goal of this workflow challenge was say that, okay, if we were given an end-to-end surgical video of this procedure happening, can the machine be taught to segment in real time? So when I say segment, here I mean like a temporally segment in real time, what is happening when during the procedure. Essentially to say that, okay, from 0 to 5 minutes, the patient was being prepared, and 5 to 7 minutes, the callow triangle dissection was happening, the clipping and cutting was happening from 7 to 20 minutes or whatever, right? So you essentially want to know if you can teach the machine to do that. So why would you want that? There are a number of use cases that you can come up with. You can come up with, like, okay, how much anesthesia does the dosage does the patient need, or even for the hospital to be able to plan that procedure. How long is a procedure typically taking? How many can you schedule per day, and so on, right? I mean, there are so many use cases that you can come up with, and which was also spoken about during the other talk that I mentioned. So again, really depends on what who you want to target. You can target the surgeon, you can come up with a bunch of use cases. You can target the hospital, you can come up with a bunch of use cases, and so on. But again, coming back to the problem itself, so it turns out that it is possible to sort of teach the machine this, and you can essentially imagine a sequence of images coming into your model. And again, the sequence is both from within the patient, and it could be a bunch of cameras that are probably, out there in the OR, wall cameras that are monitoring all of the surgical staff movement, the anesthesiologist movement, or what have you, and also all of the other sensors that are hooked up to the patient, the vitals, if there is a machine that's sort of counting in the number of sponges or whatever. So what have you, you can literally sort of come up with a state machine that takes into account all of the scene dynamics, and together with the video feed, if you are able to sort of know what tool is deployed and what is the pose of the tool, and if you are able to sort of track the tool in real time, and all of these can potentially be inputs to your model to be able to sort of segment with very high accuracy what exact step is being performed in the procedure at any given time, right? And again, I'm presenting here a paper that was called EndoNet that again came out like a few years ago. So all they did was they said, okay, they have the sequence of video frames coming in. They were actually looking at it on a frame by frame basis. So the EndoNet itself is just a VGG backbone convolutional net that takes every frame and essentially featureizes that image, essentially converts it into like a vector of numbers that are representative of the pixels in that image. And then the way they sort of add the temporal component is to have an LSTM, which essentially looks at about 30 seconds worth of images, okay, and so stitches together. So it's not directly looking at the images, but it's actually looking at the features that came out of every frame. And then when you look at it over a period of 30 seconds, and then you make a sliding window determination as to what exactly is the step that is happening. So this model can very easily be sort of augmented with other things that like the non-endoscopic feed, or oh, this is the tool that is deployed, and this is the patient vital, and so on. So all of that can make this model better, but just based on the endoscopic feed and not having any of that auxiliary information, this is still a pretty decent performance on this particular data set, where you essentially, what I'm showing here is a confusion matrix of, so this is the predicted step P1 through P7, the 7 steps that I showed you in the earlier slide. And this is the ground truth. And you can see that for most of the steps, you are at a pretty good accuracy, at least 82%. And even if there is a sort of like a wrong label, it's most likely the adjacent step. So it's not like the first step is the last step is getting labeled as the first step, or the other way around, right? So there is good consistency in the way it is done. But once again, the challenge in taking this and sort of deploying it in the OR is lack of data. So the data set that this particular paper used a subset of this Colec 80 the Colec 80 data set itself is just 80 cholecystectomy videos, all of them annotated by one surgeon. And literally, they are all so consistent that even as a sort of non-medical person, me watching like 5 videos or so, I was able to tell from the 6th video like what was going on. So it's so consistent. But surgery is often not that way. And just like with Google Maps, there's like no one way to go home. I don't think there's like one right way to do the procedure and have the right outcome. So there are procedures being done in so much diversity of techniques, and the outcomes rates are still good. So there is no real way of saying this surgeon is good or this surgeon is not. And again, the surgeon could be left-handed, right-handed. You may have to take into account something specific to the patient. Maybe they had a different procedure earlier, so maybe you have to avoid some implant and you have to do the procedure a different way. I mean, there are so many different variables here. And unless you really teach the machine all of that and be able to sort of give it sufficient diversity in the data that's being used to teach it, the model is going to end up learning a very biased approach. And the moment you plug it into a different surgeon with a slightly different technique, the workflow model is going to fail, and it's going to give you like false alarms or true negative, false negatives, or what have you, right? So really, it's a question of like, how do you scale this? And so the data awareness and also the knowledge of who actually owns the data and who has the right to give it out, and how do you sort of create a community around that so that people can start using this anonymized data without having to worry about legal implications and so on, and publish freely on it. I think that really is lacking, and that is what is kind of stopping it from going to the next step. So just for completion on the surgical consciousness workflow, so the last one is essentially on, so let's say the patient had the procedure, they went home. So again, I was at this SAGES conference a couple of years ago where there were so many talks on the use of ICG. So in this procedure, so in the step, one of the steps that I showed you, the cutting and clipping and cutting, is where they sort of remove the infected gallbladder. And more often than not, they just evaluate the patient in bright light, and they have to make a call as to where the common bile duct is forking into the cystic duct and the hepatic duct, and they have to cut here where it's going into the gallbladder and then remove the gallbladder. If they cut before this fork happens, then they could lead to serious complications for the patient, right? So there is still sort of a big debate in the medical community as to how efficient or how effective the use of ICG is. So what I'm showing you on the right is not the regular bright light image, but the patient has been administered a dye, and when you shine a light of certain wavelength on that patient, the anatomy is going to glow in a certain color, and then this gives the surgeon additional information as to like where whether they're making the right choice in cutting. So now you have two imaging modalities that are available to the surgeon: the regular bright light image, and then you have the fluorescence image. And you can very easily sort of imagine. So this is not from a medical paper, but from a sort of like a target recognition paper. But you can essentially the idea is the same. So you have a multi-stream input coming in, and you can very easily imagine a multi-stream convolutional network. And then once you sort of featureize them, you fuse the embeddings or the feature vectors coming out of those images in some space. I mean, it could be a Fourier space or any other space that you can imagine. And so that is a way you can combine an image stack, really. So an image that contains both the bright light image and the fluorescence image, and really be able to make a decision as to, okay, where really is the cystic duct? Did the surgeon, so when you're sort of analyzing the surgery after the procedure to see, okay, how well has the surgeon done? If you want to really visualize where is the cystic duct, you can sort of use this, and the machine can look at both of these images and give you a score and say that, okay, this is really how the surgeon did, and it turns out that there is sort of like a scoring mechanism. I won't get into this, but there is, this is again, something I saw at the conference where surgeons have sort of come up with this scoring mechanism where they are able to assign a score to how well the surgeon did in this critical step based on what anatomy was being visualized, how well the clearance was, and where the clipping was happening, and so on. So really, so again, this is just to emphasize that imaging can play a role in all of the steps, starting from the moment the patient walks into the clinic to the moment where you're actually looking at the how the procedure went, and you're even sort of evaluating the surgeon's efficiency. So I'm going to use that as a segue to sort of transition into microscopy. So something I use extensively at Calico. So it turns out that similar to the ICG image I showed you, fluorescence microscopy has been existing and extensively used for a few decades now, and it's really sort of is in the heart of like drug development and particularly the aging studies that we do at Calico. So what really is fluorescence microscopy telling us? So if you look at this chain here, so the use of microscopy is in what you call optical phenotyping. So just to give you a sample use case, if you're screening a patient, or rather if you're screening a particular cell line to see, okay, you have these cancer cells, what drug is going to be effective on this particular line of cells. And let's say you have to screen like 10,000 compounds. And so there is no way for us to sort of manually go through each of those compounds and record them. I mean, at least they used to be done that way, but now with automation, we really want to speed up things, and we want to be able to scan on the order of like millions of compounds to see which ones can kill the cancer cells while not affecting the healthy cells. Now, how do you define kill? How do you define healthy? How do you define sick? So that essentially comes from the scientists and the biologists who essentially tell us that, oh, if a healthy cell is treated with a compound that affects it, this is how the mitochondria changes, and this is how the nucleus changes, or whatever they know is the result of that perturbation. But on the other hand, if a cancer cell dies and the healthy cell is not affected by the drug, they have a way of saying, okay, this is how the nucleus should be, and this is the distribution of the mitochondria, and so on. Now, when you image these cells, you image them in bright light. And in bright light, you can only see so much. You can see like the outer shell of the cell. You can see a little bit inside the cell, but you cannot really make out, okay, where is the nucleus, where is the mitochondria, where is the, like the lipid droplet, or all of the other substructures within the cell. So the way to do that is to be able to sort of stain these substructures with antibodies, or today they are actually done through like genetic engineering, where you're actually tagging some proteins in the cell with a particular fluorophore. So what that fluorophore does is that when a particular gene is expressed in your body, the fluorophore, that molecule is also expressed as part of the protein. And when you shine light of a certain wavelength on it, you will be able to sort of get a glow in a particular wavelength. And so if that glow is there, that means that protein has been expressed, and that means that gene has been activated in that particular cell. Okay, so that is the use of fluorescence microscopy. So you can tag a protein associated with the nucleus, you can tag a protein associated with the mitochondria, with the endoplasmic reticulum, what have you. So these are different substructures of the cell. And each time I can actually shine a light of different wavelength on the cell, and I can extract a different substructure that comes out. And the goal of this is to essentially be able to combine this stack of images and be able to quantify them and say that, okay, all of the healthy cells had a nucleus of this size, and the pixel intensities in the mitochondria was so much. Or you can imagine a million different features that that quantify what you see, and then you can come up with clustering and say all of these features correspond to a dead cell, all of these correspond to a healthy cell, and so on. So the goal really is to be able to sort of go from microscopy images to quantifying the subcellular structures of each cell and in a population, and be able to very quickly aggregate what the image is telling us, so that the scientist only has to stare at the data. They don't have to actually stare at the images themselves. But what I'm going to talk about is not really the quantification aspect, but the aspect of how do we go about generating this. So like I said, to generate these 5 images, I need to actually shine laser light of 5 different wavelengths on the cell, and each of them gives me a different image and then different protein, right? So just like we don't like laser being shown on us, cells don't like it either. And the moment so there is a limit, a tipping point beyond which the cells suffer photo damage. And every time you shine laser light on them, it kills them a little bit, to the point where you shine enough light on it, you have killed the cell just by the laser, not by the compound. So there is always an effort in the scientific community to minimize phototoxicity or photo damage, so that you're really sort of reading what is in the cell and not the sort of external damage you're causing by shining light on it. So towards that end, one of the models that was sort of developed by Allen Institute at MIT, which we use at Calico, and Google has a model as well, is a 3D unit model, where they essentially are saying that if you remember, the original image that I told you was that there is a bright light image. So the bright light image, as in if you just shine yellow incandescent light on the cells, that doesn't do anything, that doesn't harm the cells, and the cells are happy, but it conveys very little information, at least to the human eye. But the hypothesis in this model that the MIT developed was that the information is there, just that it's not perceptible to the human eye. So they trained a model that would look at a bunch of bright light images and be able to predict what the nucleus image would look like, or what the mitochondria image would look like. Now, is it going to work on all of them? Probably not. I don't know if all of the information is available in the bright light. It does well with varying efficiencies on the different substructures, but on some channels it works extremely well. And so that basically means that the information is there in the bright light model, and you can very well train a three-dimensional model that takes as input just the bright light. And instead of us having to shine a laser light on the cell and kill it a little bit to extract the substructure, you can use this model to just predict what that substructure is going to look like, and then augment it with the others. So maybe 3 out of the 5 can be predicted, and so you have sort of reduced the phototoxicity on the cell by 60%, and you're only having to shine laser light for the other 2 structures that don't work as well. So that really was the goal of the model, to go from a stack of just bright field images, or that's a regular normal light, bright light images, and with the help of predictive models, you're able to sort of go and sort of predict what each of the different subcellular structures are going to look like, and then you put it all together, and then you go and quantify it. Then you tell the scientist, okay, you applied this compound, and this is how the mito changed, and then this is how the nucleus changed, and then you sort of make that next step in your scientific process. Okay, so and this is happening extensively today, and this really is the future. And you can see papers coming out where they're essentially doing drug discoveries completely through AI. Essentially now, we have all these generative models that have the capability to learn what different drugs are doing to the cells, and then even without even having to image them, I won't go there, but you have a completely AI based solution that is sort of screening compounds and shortlisting compounds to the point where you only have to try thousands of compounds on a cancer cell line as opposed to millions. And so you use AI to shortlist your compounds significantly. So the last slide I want to show here is sort of like that's something we do at Calico, and we are allowed to share publicly. Essentially, it is what we call, so this is what you see here, are little yeast cells, budding yeast cells. So a yeast cell, essentially like so similar to the yeast that you use in cooking, but these are budding yeast. The difference is that budding yeast lifespan is measured not in chronological time, but it's sort of measured in based on the number of daughter cells that they produce. So every time they produce a daughter cell, and then they wait a couple of hours, they produce another daughter cell, and so on. And typically, by the time they produce about 30 daughter cells, they die. Okay, so that is sort of the typical lifespan of a yeast cell. So what we have here, these are little traps. Okay, so the little kidney-like things are little traps, and they are trapping a yeast cell, and then so that it cannot escape, and then it keeps budding, and the buds keep going away, but the cell keeps budding, and then we are able to monitor until what time the cell is alive. Okay, so this setup is completely automated using computer vision, and so you have the microscope automatically taking images every 10-15 minutes. It could be a bright light image, it could be a fluorescence image. We have a way of sort of segmenting out the traps, we have a way of segmenting out the cell, we have a way of knowing when a daughter is budding, and then we have a way of literally counting how many times the cell has bud before it stops doing so, to be able to generate what we call a lifespan curve here. So the goal of this lifespan curve is to say that, okay, a normal cell would, so this is your probability, the y-axis is essentially the probability of being alive. This is the number of buds that the cell has. And a normal cell would probably follow this blue curve, where typically after about 40 or 45 buds, the cell has a very low probability of living. And you can imagine mutations or mutants that you can create, where you say, oh, this is a short-lived mutant. Maybe I have another mutant which is longer-lived. Or maybe I can just change the media in which the cell is living. Now the cell is happier. Maybe it creates more buds, it lives longer, and so on. So again, the goal of this slide really is to illustrate the capability of like being able to scale and both automation and throughput using computer vision, so that the scientists don't have to sit and watch this, and they are able to just set up an experiment and then go home for the weekend, and then they come back. You have like close to like 1 million cells, or how many ever captured, and then the machine can automatically segment it, and all you have to look at is a curve, and then so you can take that next step in your scientific experiment. So again, so that brings me sort of to the end of my talk. But really, the whole goal of the talk was to sort of showcase the importance of vision in applications that have a wide range, but also sort of showcase the sort of lack of diversity in data that sort of hampers medical data today. It's not so much in the biotech community, but even there, I wouldn't say that the data sets are as rich and diverse as, let's say, like the self-driving community and so on. But really, the whole point was to sort of illustrate what vision can do. Thank you.
Thank you very much, Jagadish. Great talk, very fascinating. I want to open it up for questions, so everybody please feel free to send in your questions, and I will relay them. Until in the meantime, I have sort of more of a technical question. So you were showing before the Unix architecture, right, with these attention blocks and the dense sort of architecture compared to the what you talked about later on, like you mentioned for the label-free prediction, you're just using a straight-up 3D unit. Similarly for action recognition, right? So there's the EndoNet, which is basically just straight up, a CNN LSTM, and then right on top there, this more involved system, right, trying to detect hand movements and object detection and scene analysis, and then feed those all through a Markov model. I think you have it there. Just kind of curious from your experience. So, how much of your work is sort of devoted to trying out these various architectural bells and whistles, and how much is do you find that getting a good or a stable result is more a matter of sifting through the data and visualizing the failure modes and that kind of thing? Sort of how do you find your best operating point is for balancing those two kinds of efforts?
Good question. So my main objective is to solve Calico's problems. It is not necessarily to come up with the like next Mascar CNN. I mean, if it happens along the way, I'm happy. But so I don't want to overcomplicate the model that I throw at my problem. If a simple unit can give me 95% accuracy, and if that can already sort of tell the scientists or inform the scientists of the logical next step that they have to take in their experiments, be it so. But if it so happens that the model accuracy is lacking, and then you have to come up with innovative ways to sort of work around that. So the first thing, we also explore changes that can be done on the data side, because if you have extremely good data, even a naive model can do really well. And I think most people understand that. So the strength is really in the data. So you can throw the kitchen sink at bad data, and then you're just going to get garbage out. So I think the quality of annotations and the quality of the data really define what you want to throw at it. But despite that, if you have like hardware limitations or time limitations and so on, you say, okay, this is the quality of data I'm stuck with, and this is the kind of annotations I'm stuck with. Then we start going and exploring. Okay, can we think of ensemble models? Should we come up with different ways of combining this and sort of creating various cross-validation data sets, and what can be done? So, and I think that the more precise answer to your question, it sort of depends on the specific application. So segmenting yeast cells compared to segmenting, like the spine, maybe completely different beasts, and you may be solving for different problems. And so in one case, it may be okay to have a few false positives as a bud, but in another case, it may not be okay to have even the slightest false positive in terms of a gallstone. So I think the way we approach it is essentially to sort of study the problem, understand the data, and then sort of research off-the-shelf solutions that are out there first.
Yeah, I totally agree. It always starts by looking at the data and looking empirically how things behave.
Yep.
Yeah. So there's a question here about. Curious to hear your take on this as well. On in robotic surgery and the importance of haptic feedback versus just using the visual interaction. How much do you think this is critical?
So I mean, I only heard I only have second-hand information on that. I haven't sort of directly spoken to surgeons about it, but essentially what I've heard is that like you find that when you're using laparoscopy, when you're doing open surgery, you can actually sort of feel like the amount of tension in your suture, or like how much you're pulling and so on. Whereas with robotic, where the movement is completely in the air, and so you are sort of moving your fingers as if you're making a knot, but how tightly you're pulling, and whether you're actually damaging anatomy in pulling too tightly, or whether your knot is not as tight as you think and it may come off later, creating a complication, is sort of something that the surgeons grapple with. And the only thing they have to go off of to determine how well they did is what they see on the screen. If you essentially have a tight knot and you can see that, oh, the skin is, the tissue is sort of coming together and looking like it's tight or has a sharp edge or something like that. So those are the kinds of cues they sort of go off of based on their experience. But I at least for one, definitely feel that if there was a way of sort of mimicking haptic feedback, I think it would definitely go a long way to enhance their experience.
Yeah, because it's a whole other new skill set to learn once that's gone.
Absolutely.
When they transition to robotic. Great question here from William about the diversity of data that you mentioned in the context of surgical intelligence. So the question is basically, this seems to be a general problem in AI. You can't feed it every single input to learn perfectly. Is there a way to quantify when the learning set is diverse enough? Right, so how do we quantify this beyond saying, okay, we're going to cross-validate, but even if we're cross-validating, right, many times we're setting aside in our test set only a negligible number of examples if the data set is diverse enough. So any thoughts on how we can really quantify diversity and sufficient diversity in the data sets in a powerful enough manner?
Great question. Great question. So this is something we think about a lot even when we want to give away code at Calico, right? I mean, let's say we have developed something like this yeast pipeline that I was showing you, and we say that okay, so it works really well, and it seems to be informing the scientists. Let's go ahead and give out the code, right? And then the next lab comes along and they plug it in and it doesn't work. So so the thing is, like, I don't think any one entity can really have or capture the diversity of data that is out there and come up with a solution that solves everything. And I don't even know if that is the right approach. Instead of having one average model that performs with 70% accuracy on all data, is it better to have customized models that perform with 90% accuracy on small data sets, but which can be taken and transfer learned or tweaked for different data models? Right. So I think the way we think about it when we want to give away code really is that, okay, I'm giving out a framework. Okay, I'm giving out a framework and I'm saying that on this data set, which was collected with X, Y, and Z parameters, this is how it works. If you know how your X hat, Y hat, Z hat differ from X, Y, and Z, do the appropriate pre-processing, but you can very well use my framework and sort of, instead of having to start over, the technique is out there. So your starting point is much higher than it was when we began. So that's kind of how I think about it.
Mm-hmm. You guys using in microscopy like domain adaptation or just straight up transfer learning?
So for at least for microscopy, I think at Calico there is not much of a paucity for data. I mean, it's very easy to create like yeast images over the weekend, sort of train our models from scratch. But we do sometimes do transfer learning off of like the Broad at MIT. They put out these models that are trained on like public data sets of yeast cells or human cell lines, and it's very much easier to sort of transfer learn off of that. Yeah.
Yeah, this is a question we get a lot too. Considering variations in patient anatomy and vendors and quality of images, how many imaging samples you need to develop a valid result or model? So yeah, the answer, the short answer for this is always it depends, and maybe the long answer is if you have reasonable control, then proof of concept for segmentation, a few hundreds of examples. You need to define what that means, and more production level system, it's going to be thousands. But curious in your thoughts, Jagadish, on this as well. How do you usually answer this question, or how do you define the ground rules for starting to address this kind of question?
I don't know if I have a good answer. So because the most sort of publicly used data set that I ever come across is this UK Biobank data set that we at Calico also extensively use. So the UK Biobank, to my knowledge, is sort of the best repository out there of 500,000 individuals who volunteered themselves for research, and they have been sort of imaged and phenotyped from head to toe. They can find like all kinds of like ultrasound, cardiac MRI, like all kinds of data. But even there, you run into the problem that because it is such a consistent, clear protocol, all done by one entity, the images are so consistent that you can very easily create a model, and you see like tons of papers published off of this. But how valid is that? And again, it's dominated by Caucasians in the UK and demographics and so on. Now, if you want to take that and generalize to like the Indian population or the Asian population, like how easy is it to do so? And whether the X-ray and CT images coming out of equipment in those countries is going to match the quality and consistency of these images is definitely up in the air, right? So again, like I said, I think the goal really should be to be able to take something and then customize your solution. I don't think any one company or any one entity can solve this.
Yeah, it sounds too good to be true. Such a huge data set, but there's always going to be a catch.
Yeah.
Another question, more general question by Joel: is there a limit to automated computer vision algorithms in terms of voxel size, pixel size, frame rate, DICOM frame rates by modality? So I mean, the direct answer to this question is it depends what you want to achieve, right? So if the frame rate is so low that the motion information is not there, then you will not be able to achieve it. But there are no hard limits on this. It's just you're going to be able to extract the information that's reasonably present in the data you're looking at. I don't know, Jagadish, any thoughts on your end on this question? Limitations and resolution and frame rate and the like.
No, I agree with your answer. I was going to say the same thing. Yeah.
Okay. Yeah, Joel says thank you very much. One more question I had about, again, going back to this vision of surgical intelligence, this more far-reaching vision, and you were saying how the limits on data availability are really playing a part in the challenges facing this field. Nevertheless, just from your experience in the space, where do you think the applications are going to be most ripe for implementation in the field? Is it going to be intraoperative, applications like a prediction of the next step, or is it going to be easier in preoperative planning or an analysis of surgeon performance? Where's sort of the low-hanging fruit in terms of having a relatively quick pathway to production and to actual implementation?
I think the low-hanging fruits are definitely in the pre-op and the post-op, the sort of non-real time where it is not directly impacting the patient. Well, the pre-op does impact the patient to some extent indirectly, I guess, in the decisions that they make as to what is the further future course of action. But definitely sort of a real-time approval from the FDA is something I think every company is dreading. And, and it's not really possible to sort of prove the use of deep learning in every possible scenario or use case. And I'm not even sure if the FDA knows how to evaluate a deep learning algorithm in real time that is affecting the surgical decision making, right? So I think that there is definitely some sort of room to go there, in my opinion. Yeah, so definitely the low-hanging ones are pre-op, and also there is all this stuff associated with analytics, where essentially not directly related to the outcomes, right? I mean, in terms of the number of tools you're using, and just inventory, and there's already a lot of things happening in that field, but that also vision could play a role.
Mm-hmm. Yeah. On the other hand, whoever does have access to the proper data is obviously going to gain a significant advantage here.
Yeah.
Because eventually it's going to go to the real-time environment itself, and probably depends on what type of application is being done. Like, very much depends on what people are going to try to claim in terms of the usage of this, depending on how it's formulated. One more question from Sirius. So the examples provided was from CT and microscopic images, which are still images. What about echo data? Yeah, so echo data is always going to be a video. It's always going to be important to take that information into account. So typically you won't only be using frame-based classifiers, but you'll eventually try to teach the network to use the temporal information as well. Within those videos, also to detect, the proper part of the clip to use, and various other applications. There's no inherent limitation here to still images, whether 2D or 3D. I think Jagadish showed for surgical video one of the architectures that's used for more temporal kind of data. But yes, absolutely, the time domain in echo cannot be ignored at all. That's sort of my take. Jagadish, any thoughts here, like on echo versus the more still modalities?
Yeah, I think definitely like echo and to some extent like the MRI, cardiac MRI images, for example. I think that definitely you can't just look at one still frame and sort of make a determination on, is the patient sort of the pumping rate of the heart normal, or is there like it's slower than it is, and things like that. So you can actually look at the sequence of frames and you can literally watch it. And if you have segmentations on a per frame basis, by just looking at how much the ventricle is contracting and expanding and so on, you can make determinations that are clinically meaningful. So I think there are definitely a few modalities out there where I think a sequence of images plays a critical role.
Yes. Okay. I think we'll wrap it up here. We're it's already 2:30 p.m. You know what? I see we've got still a large number of people in here, so maybe just one last question. And thanks everybody again for the response and for the interaction. So just one last one, and then we'll wrap it up. Which fluorescence enzymes are used to tag cells? Are multiple used at once, and how do they affect the model? That might be out of scope, but go ahead, Jagadish.
So I'm not an expert in this certificate, but I can point you to like these fluorescent proteins called GFP, YFP, and so on. So these are called green fluorescent protein, yellow fluorescent protein. There is cherry, and so the colors basically refer to the spectrum of light which excites them, and then they come back in a different wavelength. So you'll get like something in the green wavelength, something in the red wavelength, the cherry wavelength, or what have you. So depending on the particular protein or particular gene that you want to tag. So I'm not an expert on the biology side, but the systems are extremely advanced that you can actually go tag a gene with this particular molecule of a desired color. And so the mito can be red, the lipid droplet can be green, the endoscopic reticulum can be blue, so that you have very minimal overlap between this, and what you're visualizing truly is what you want to be seeing.
Thanks. Yeah, and if you guys are getting some of this functionality to work, you know, without the dyes as well as you showed down there, then that's quite amazing.
Yeah.
Okay, so thanks again. Thanks Jagadish for a great lecture. It was really a pleasure to host you at our meetup, and thanks everybody for attending. And we'll stay in touch and let you know about the next events as well. So have a good rest of your day, everybody.
Thank you, everyone. Thanks, Moshe.
Thank you. Bye.